What's up, guys? This is my last minute biology video, and in this one we're going to be talking about uh, mostly about viruses and about what those are and about what viroids and uh, pyrons are and prions, sorry. And we're going to go ahead and take a look at that. So one of the first things you need to know about a virus is what it is exactly, and it's not a cell. All right, that is very important. So in terms of viruses, they're not cells. Okay, and that's very important to know. It's only alive, quote unquote, when it's inside of another cell. All right, so like you're gonna have this nice little bacteriophage, and we'll get into that. And it's basically a walking around, and it's not living, but it's going to get attracted chemically to a cell. It's going to enter the cell. It's going to start replicating its DNA and producing more of itself, and then it's going to go out, and those reproduced bacteriophages are going to go into another cell. So they're only alive when they're within a cell, and I'm not exactly sure why they categorize them like that, but that's important to know that they're not living, and they're not a cell. Okay, so you're also going to want to know what the structure of a virus is, and they can look like bacteriophages. For right now, we're just going to use a circular diagram of it. And basically, what you have a DNA strand, basically, and that's going to be surrounded by a protein coat. Okay, and so this is going to have like a protein coat around it. And what that protein coat does is it allows the virus to kind of take shape, and it's also called a capsid, right? So this kind of like cell wall, basically, for the virus is called a capsid. Now, basically what happens, I believe, I'm not quite sure, is as this virus enters a bacterium and then it leaves, what's going to happen is as this bacterium leaves, it's going to take part of the lipids from this cell membrane, and it's going to use that to make a covering over the capsid. So the virus is going to replicate inside the bacteria, and it's going to leave the bacteria, and as it leaves, it's going to take some of the lipids from this cell membrane, and it's going to make something called a glycoprotein receptor. And what that is it's basically just something that looks kind of like this, and it basically helps it find the right cell. So this is going to fit on to the correct cell, and that's going to allow the DNA to get injected and then duplicate and then make more of these tiny red viruses that will then go and infect another cell. Right? And those are called receptors. So, alright, let's go ahead and take a look at a bacteriophage and what that is. Kind of the generalized way of looking at bacteriophages are you have kind of this pentagon looking structure at the top, and it's going to go down into what's called the tail, and then you're going to have kind of a square at the bottom, and you're going to have these legs coming from it. Now those are called short tail fibers, and sorry about that, let's scroll down. So these are called short tail fibers. This is called a base plate. That's the tail, that's the head. But basically what happens is you have the DNA inside of, this bacteriophage. And what happens is bacteriophages only attack bacteria. They're viruses that only attack bacteria. And what happens is it brings us to the lytic cycle, which is spelled Lytic cycle. Now, what this is it's basically the nature of a virus. It's what the virus does. So we're going to have a yellow cell, right? And this cell is chilling. And what happens is this bacteriophage is going to come up to it, and it's going to attach on to the cell, right? So that's not good. No one wants that, but it does. And what happens is it injects its DNA into the cell. That is the first step of this lytic cycle. The next thing that happens is the DNA gets replicated and transcribed onto the basically into messenger RNA, and it kind of incorporates itself into the cell's DNA. So what's going to happen is you're going to have the cell, and you're going to have the bacteriophage, and it's going to be in DNA, and it's going to move over to messenger RNA, I believe. And then it might like fit part of itself. All right, it's probably going to transcribe itself. This is going to be the cell DNA. It's probably going to transcribe itself onto part of the onto the cell DNA. That's what I understand this kind of to be. Now, what happens in this next step is you're going to have the same cell, right? And what happens is a virus is like a factory, right? It's basically what's being produced, and the cell's like the factory. So it's going to put all of its parts into the cell, and the cell is basically going to reproduce it. It's like a factory. So what happens is the messenger RNA that was created in the second step are going to be processed into proteins. So what happens is this the messenger RNA that was created after it's transcribed on it's going to be made into proteins, right? Because as the cell performs protein synthesis, it's going to read along and make its normal, enzymes and proteins and all that good stuff. And then it's going to come across the virus's messenger RNA, and that's going to make it so that the cell starts duplicating the virus as well. And what happens is, I believe it's polymers is used to do this. And what happens is, basically, the cell starts creating the different parts for the virus. So you're going to have the legs, and then you're going to have the base plates. And all of these things are going to be floating around here as proteins. Now the next step, as you can probably guess, are these proteins are going to be put together by the polymers, and they're going to leave. And this is called lysing. And what happens are basically the final step is you're going to have the cell, and you're going to have the bacteriophage, many bacteriophages, and they're going to basically push up against the cell membrane until they leave. And it's kind of going to burst like that, and they're all going to leave out that entrance, and that's called lysing. There is a different cycle called the lysogenic cycle, where what happens is you have the first two steps. So the virus injects its DNA, it becomes part of, I guess, the cell genome, and then what happens is you're going to have the cell, then you're going to have the virus DNA, I believe, then you're going to have the rest of the cell's DNA. And that's kind of what happens, I believe. And as the cell duplicates, it's going to duplicate the virus DNA as well. So you're going to have all of these cell divisions, right? So this cell is going to, split in half, and then you're going to have part of it as the virus DNA and another part of it as the cell DNA. Now what's going to happen is it's going to duplicate, and then those cells are going to duplicate and duplicate and duplicate and duplicate. And what happens is occasionally a prophage is going to leave the cell. So I guess basically that's just the virus is going to leave the cell and do this to another one. But for the most part, these will usually stay dormant. And an example of this is kind of like chickenpox. Chickenpox can come back as shingles, And that's what happens is chickenpox, you get it, you get better, but these cells lie dormant, and occasionally they'll mutate, and then they'll kind of emerge from this dormant state, and then you'll get shingles from it. And that's how all of this works. Two really quick things are prions and viroids. And a viroid is basically a RNA without a protein coat, right? And so that's the important thing to know is it's RNA without a protein coat. It's kind of like a virus without a protein coat. Prions are infectious proteins, right? So what happens is you have this sort of protein, I guess, and this protein kind of gets mutated from its original form, and as other proteins come in contact, so let's say we have a same type of protein, but I'm going to represent it in a different color. So this protein comes up to this, it's going to then become mutated, and it's basically an infectious protein, and it's not good, and what happens is it gets converted into some other configuration. So I hope that helped, guys, and have a good day.